Welcome learners. I am Dr. P. Venkatamana from the discipline of Anthropology, School of Social Sciences, IGNOU. Today we will be discussing about the practical manual of one of the core courses, namely the Human Population Genetics of the program BSc Honours in Anthropology. This program, BSc Honours in Anthropology, has 14 core courses and 4 discipline-specific courses. Each course has a practical manual. Here, the student should know that the study centers are not conducting any practical sessions, and there is no practical examination just based on the practical manual. However, the questions would be warranted from the practical manual, which means the questions would be asked from the practical manual in the term and examinations. Because students are asking, would there be any practical sessions and practical examinations based on the practical manual? So there is no practical sessions and practical examinations. Only it would be practical questions. Questions would be warranted from the practical manual in the term and examinations. Now, so coming to anthropology. What is anthropology? Anthropology is a holistic study of human being, which has 4 main branches: social, cultural anthropology, physical, which is also called as biological anthropology, archaeological anthropology, and linguistic anthropology. So, coming to physical anthropology or the biological anthropology. This is one of the main branches of anthropology, this physical anthropology. In early days, the physical anthropologists were trying to understand the human variation and evolution based on metrical characters like anthropometric characters, which are of polygenetic in nature. But in 20th century, scientists were discovered some of the genetic characters where we can calculate gene frequencies. So, this population genetics is a component of physical anthropology wherein the scientists can calculate gene frequencies because these gene frequencies would be used in understanding the human evolution. Basically, the core aspect of physical anthropology is to understand human evolution and variation. The variation is based on, like anthropometric characters, blood group polymorphism, and recently DNA variation. Like the way the human evolution is studied with the help of the gene frequencies that we could able to calculate for some of the genetic characters like ABO blood group system, RHD blood group system, and phenyl PTC testing sensitivity test, and recently DNA polymorphism has played an important role in understanding the human variation and evolution. Now, coming to some of the genetic characters that we have been discussing in this course, like blood group system like ABO and RHD blood groups, and color blindness, and PTC testing ability test. So, these 3 tests that I will be discussing in this video, you may expect questions in the term and examination based on these experiments. Now, coming to ABO and RHD blood group system. So, what is this ABO and RHD blood group system? This ABO blood group system, this ABO blood group system has 4 blood groups: A blood group, B blood group, AB blood group, and O blood group. However, Karl Landsteiner discovered A, B, and O blood groups of ABO system. But De Castello and Steiner, these are the two scientists, they have discovered AB blood group of the ABO system. However, Landsteiner and Wiener have discovered RHD blood group system. Then a brief description about the blood group systems. So this A blood group has A antigen and antibody B, and A blood group persons can donate blood to blood group persons A and blood group persons AB, and can receive blood from blood group persons A and blood group persons O. Coming to B blood group, which has antigen B, and in the same case the B blood group has antibody A. This B blood group persons can donate blood to B blood group people and AB blood group people, and can receive blood from B blood group people and O blood group people. Coming to AB blood group. So in this case, this blood group has both antigens, A antigen and B antigen, and it doesn't have any antibodies because it lacking any antibodies. However, this AB blood group persons can donate blood to only AB blood group people, but can receive blood from people of all the blood groups like A, B, AB, and O. And O blood group people it doesn't have any antigen, and they have both anti-A and antibody B, and can donate blood to all blood group systems and can receive only from O blood group. Then coming to how to detect how to perform the blood group typing in the laboratory. So for that we need to prepare normal saline. How to prepare a normal saline? Take 8.5 grams of NaCl and add into 100 ml of distilled water. Make it ready, this normal saline. Then we have to make 3% red blood RBC solution. Then we have to perform the experiment. Then how to make it? We have to collect 2 ml of blood into the test tube, which is already having anticoagulant. Then centrifuge this at 2000 rpm for about 1 or 2 minutes. Then take out the supernatant, which is on the upper portion of the red blood cells. Then add 2.5 ml of normal saline into this blood, and then centrifuge it. It is 3% red blood cell suspension. Then take 4 small test tubes or a porcelain tail. So the blood group system can be performed with the help of 2 methods. One is porcelain tail method and the other one is test tube method. In the case of test tube method, what we have to do is first put 2 drops of this 3% red blood cell suspension into every tube. There are 4 tubes. I said in the beginning there are 4 tubes: A, B, H, and D. Then add already commercially available anti-sera, anti-A, anti-B, anti-H, and D. Add 1 drop of anti-sera A in tube A and anti-sera B in the next tube, anti-H in the other tube, and anti-D in the next tube. In some cases, you may not require anti-H. In that case also, we would be able to distinguish between the blood groups. So what we can do is, so after that, once we have added the anti-sera into the 3% red blood cell suspension in the case of test tube method, we have to centrifuge them gently, maybe at 1000 or 2000 RPM. Then take them out from the laptop, then watch for the agglutination reaction. That is number one. Number two method is slide method, wherein just take a porcelain tile. There are 4 grooves, 1, 2, 3, 4. Add 3% red blood cells mixture in 1, 2, 3, 4. Then add the commercially available anti-sera. In the first groove, add anti-A. In the second groove, anti-B. And the third one, anti-H. And the fourth one, anti-D. And if you don't have H, doesn't matter. Then see for the, then gyrate this, this porcelain tile gently and cover it with porcelain tile for some time, a petri dish. Then watch for the agglutination reaction, if the agglutination reaction is happened or not. Then we can interpret the results like that. Suppose if there is an agglutination present in test tube A, and if there is no agglutination present in test tube B, and if there is no agglutination present in test tube H, we can come to a conclusion that the person belongs to A blood group. In the same case, absent in anti-A and agglutination present in anti-B and absent in H, and this is B blood group. And if the agglutination is present in both anti-A and anti-B and there is no agglutination anti-H, the person belongs to AB blood group. And if we come to the next one regarding O, if there is no agglutination A and no agglutination B, if the agglutination is present in H, the person is O blood group. In the same case, anti-H, if there is no agglutination anti-D with anti-sera, the person belongs to RH positive, and if there is no agglutination, the person belongs to RH negative. Then, so like that we can perform the ABO system. Then the next genetic character that we are studying in this practical manual is color blindness. So majority of the color blind people can see colors except red, green and blue. So this color blindness is nothing but color vision deficiency. So which means but a normal person can see all colors except red, green and blue. So it is a genetic character. So it is of 3 types: monochromacy, dichromacy, anomalous chromacy. So we will explain one simple inheritance pattern with the help of this diagram. Say for example, a normal woman married to a color vision deficient male person, he will have 50% of males with normal vision and 50% of daughters with carriers, which means the daughter may not exhibit that she is having color blind, but she is a carrier. If that daughter got married to a male with normal vision, they will have 50% males with normal vision and 25% daughters with carriers and 20% sons with color vision deficiency. So, how to perform in the laboratory? How to detect whether a particular person has a color vision deficiency or not? So, for which there is a experiment with the help of Ishihara chart. There is a chart called, this is the chart. So, this chart has 38 plates, 1, 2, 3, 4, like that, 38 plates. And the plate numbers 1 to 25 are for literates, and for the plates 26 to 38 are for illiterates. The first 21st plates help to distinguish normal vision individuals from red-green blood individuals. From plates 25 to 22 to 20 intend to separate color vision individuals so detected into protonomia, which means if they are already detected some kind of color vision, these three plates like 22, 23, 24, 25 would distinguish the color deficient people. So, how to perform this experiment? So, these are the charts. So, we have to ask the subject to sit in a well lit room, place the charts in front of the subject just 75 centimeters away from him, then ask him to read the numbers. If a subject is able to read perfectly till 30 numbers, we can consider them as normal. Suppose if he is not able to complete, if he is able to read till some 14 to 17, we can conclude them he is color vision deficiency. But however, in the case of 26 to 38, so this is for illiterates. What we have to do is, we have to give them a kind of pencil or pen. We have to ask the subject to draw the winding lines. There are zigzag lines. Whether he is, see, we have already the standard chart. We have to compare whether the person is drawing according to standard one or not. Depending on the chart, we can conclude whether the person is red blind, green blind or red blind. So, this is the color chart, this one. Based on this, we can conclude a person is having color vision deficiency or not. So, before then coming to this one, so this one, the plate number 1 to 13, if he is able to read perfectly, suppose normal vision person, this is 12. He has to read 12. Plate number 1 is 12 and plate number 2 is 8. Suppose if he read some other number, so in case instead of 12, he reads some other number, we can conclude that he is having a kind of red-green color blindness. Same in the case of, this one, illiterates, if they are not able to draw the winding lines according to the lines that are present in the diagram, we can conclude them whether they are colorblind or not. Say for example, if he is able to write zigzag way, not other than the lines that are as in this standard one, we can conclude them. So in that case, we have to repeat the experiment multiple times before coming to a conclusion. So based on this test, this is one of the important tests for the genetic characters where we perform in the laboratory. So with this we understand the genetic variation and evolution based on the alleles that we have calculated collected from the populations. The third one is phenylthiocarmide test, or it is also called as PTC tasting ability test. This test was based on the threshold value which was given by Harris and Kalmus, and this is also a genetic character which distinguishes people from tasters and non-testers. So it is controlled by a pair of alleles, capital T and small t. Capital T is dominant allele and small t is a recessive allele. And capital T and capital T, small t are testers, and both, if a person is having both small t, and they are non-testers. So to understand population variation, or if you want to understand the evolution of the human being, this is one of the tests that we have conducted earlier to calculate gene frequencies, how populations have been evolved from older generation to Homo sapiens. So how the technique is performed? There is a procedure. So we have to take 50 grams of phenyl thiocarbamide and add into 1 liter of distilled water, tap water. So we have to mix 1300 milligrams of PTC solid into 1 liter of tap water, and stir it thoroughly. This is solution 1. From there, from solution 1, take out 50% and add. This is the solution 2. So like that, you have to make solutions up to 14, which means the concentration would be reducing. The solution 1 may have more concentration and the solution 4 may have less concentration, because you keep on adding a liter of tap water into the 50% of the first solution. From first solution to second to third one, 50%, like that. You keep on increasing the concentration would get decreased. Then, so how to perform the test? We have to ask the subject to sit in a room. We have to make 14 tumblers, and then we have to ask the subject to taste the serial numbers from 14 to 1. So he may taste at one point, say for example, he may taste at 9, or he may taste at 8, or he may taste at 7. So whenever he started tasting this tasting ability, then we have to repeat the test whether he is really having a taste or not. So once, if the taste is bitter, we can conclude that this person is having threshold value of 8, 9, 7 or that. Many people may not have bitterness till 6 or 7 numbers. However, some people may not have bitterness even at the solution 1, which means they are non-tasters. And some people may have bitterness at solution number 10 or solution number 9, which means they have the alleles or genes which are for tasting ability. So like that, we can test the tasting of the person based on the cereals. So other than this PTC test, there are various tests to understand the genetic differences between the human populations. Say for example, anthropometry. Anthropometry is nothing but measurement of the human beings, which is influenced by multi-genetic nature. So we may not able to calculate gene frequencies for anthropometric measurement. So scientists have devised some characters where we can calculate gene frequencies. With the help of gene frequencies, we can see the human evolution. And how to understand the human variation? The human variation may be with the help of anthropometric measurement or cranial measurements or some kind of dermatoglyphic measurements, or in case of blood group systems, or in case of recently a DNA variation, etc., etc. But the more recent technique is DNA polymorphism. With the help of DNA polymorphism, we can conclude that these people are definitely susceptible to some kind of diseases. So, for example, when we go for diabetes mellitus or hypertension, whether these are of genetic or nature, and the scientists are still struggling whether the diabetes mellitus or hypertension are of genetic in nature or environmental in nature. Even the cancer, once we detect the cancer, this particular gene is capable of having any environmental effect or not. So scientists are struggling to understand whether genetic in nature or environment has played an important role in understanding the evolutionary variation of the human populations. So based on some of the many characters, we can understand the human population variation. Thank you so much.